have two students working on this project, Reagan and Shaquille, and our questions relate to accurately diagnosing rose rosette virus in infected plants, where the question being what is happening in a plant when you have a portion of the plant that shows serious rosetting disease, and on the other part of a plant you may have healthy looking branches. Is virus equally distributed or not across the plant? And how do you perform sampling and diagnostic testing? Our infectious clone technology does not have GFP in it. So while we can effectively deliver virus, tracking it without GFP could mean that we could sample in the wrong location, identify false negatives. So we wanted to develop a technology that we could do while trying to advance GFP technology that would lead to accurate diagnostics. And so, so thinking about this in what we understand about how viruses move really comes from studies primarily conducted in tobacco plants, which is just a vegetative plant with a single axis. And going back from 1934, our models of how viruses spread in plants have not changed, where the apical dominance drives virus movement from the inoculated leaf down into roots and up into the youngest tissue. And then the viruses fill in sink tissues and then source tissues over time. But considering roses have a completely different lifestyle from tobacco plants, and we really had to ask these questions of where is virus moving and how is it moving? What kind of systemic signaling is important for growth and defense in plants? And so, as I mentioned, tobacco and other plants have a monopodal inflorescence with a primary axis that is always a primary axis and lateral positions along that plant. But if you think about roses, they have a sympodial lifestyle, which means the apical dominance can change from one branch to another. You can get growth from the bottom of the plant rather than just the top of the plant. And so sampling and understanding how viruses distribute through the plant may not be exemplified by typical textbook studies that have been conducted until now. So the question that we're trying to answer, I think, is important for breeders and for diagnosticians in understanding how growth patterns can affect virus detection and virus spread throughout the plant. Are there potential regions where virus never enters, or does it take a longer time to enter certain regions of the plant? If you were to treat a plant for disease, what do you do? So Star Roses has been wonderful in providing us with rooted cuttings of plants, and our model system is really developed off of their supplying us with rooted cuttings, which we're extremely grateful for. And we're beginning to ask some of the certain specific questions where the rooted cuttings we get from them have a primary axis that's developing off of the lateral bud from the rooted cutting, and then additional lateral buds may spring up, and they can change in dominance over time. But with the rooted cuttings, they become an effective model plant for plant architectures. And we're asking, does virus follow the route of photoassimilate movement from shoots to branches and roots? And as the growth patterns change, does the virus distribution change? Not easy questions to investigate, but the way Reagan has been going about it was to take populations of plants and divide them into different architectures, where some plants have a single shoot, some plants have two shoots, some plants have three or more shoots coming out of the basic plugs. And then using carboxyfluorescein dye to track phloem connectivity between different shoots has helped us to try to predict where virus is moving or restricted in these young rooted cuttings, which would just be a wonderful way to help us develop screening technology for resistant germplasm also, if we can do this type of tests in rooted cuttings. And so using carboxyfluorescein dye, we attach it to the petiole of an excised leaf, and that delivers it into the phloem where it tracks throughout the plant and follow that dye tracking over 24 hours. And you can see that where the asterisk is where the dye has been applied. It moves within 5 hours to the youngest tissue, and then over time it starts to fill in tissue in between, as predicted even with a plant with a monopodal lifestyle. We found that when we inoculate virus into leaf, leaf 3, say, we can detect high levels of virus in leaf 4, or in leaf 2, a little bit less in leaf 4, and then also in the top leaf. And then over time, we continue to track how it fills in, and we found that 11 out of 25 plants, or 13 out of 25 plants over a 3-week period would produce positive results in the leaf above and below the inoculated leaf, with fewer leaves showing positive at the top of the leaf, at the top of the plant lid at 3 weeks post-inoculation. And then in a more complex architecture, such as here, we've got a single main stem with upper branches. Here we've got bifurcating lower stem, and then on the right side, you've got a lot of branches also coming off the main stem here, and understanding which one is your dominant stem is really important for understanding how virus is tracking. None of this seems particularly straightforward considering the growth habits. When we apply dye, so here on the left, we apply dye into this single stem and it goes directly upwards. If we apply it to a plant that has a bifurcating stem, it seems that the dye stays within the stem that it was applied to. And over 24 hours and longer, it never moves into the other stem, not particularly well. In this example, where we've got a single stem with lots of branches, the dye doesn't seem to move again into neighboring branches and seems to be contained in that branch. And so based on that idea, we would expect perhaps if the virus is following the path of the dye, it may also be restricted in the same tissues that the dye is restricted, which would be great to know if we can use a dye to make these predictions of where to sample. So when we inoculated plants with a single shoot, we inoculated plants with 2 shoots, we inoculated plants with 3 shoots, and then we sampled the leaf below the inoculated leaf, the leaf above, and the top leaf in the same shoot, as well as roots. In this case, at 3 weeks and at 9 weeks, we're able to detect 100% of the plants showed up positive in the upper leaf, in the top leaf, with distribution throughout the other leaves. If we had plants with 2 shoots, 6 weeks gave us the best results across all sampled tissues, in fact. So at 6 weeks, we were able to get 100% detection in the lower leaf, the upper leaf, and the top leaf, both in the inoculated shoot and in neighboring shoots, which was surprising and good to know, new information. So if we go over to plants with 3 shoots or multiple branches, we looked at the inoculated shoot, the neighboring 2, 3, and 4 shoots. And at 6 weeks, we're able to see virus distribution across each of the shoots. In the neighboring shoot 2, 3, and 4, we just decided to study the uppermost leaf, the youngest leaf of that shoot. And you can see at the top leaf at 6 weeks, we get 73%, 71%, 72% of them testing positive. And then at 9 weeks, we get up to 80%, 60% of the top shoot. So there's some fluctuation in levels. If you look at shoot 3, at 3 weeks, it goes highest at 6 weeks, and it seems to decline a little bit at 6 weeks, which represents the unpredictability, which based on all of this, my recommendation would be for sampling to take the top leaf really at the earliest 6 weeks from an infected shoot and neighboring shoots in order to get the best possible prediction of outcomes. The roots are also highly reliable, where we get 90% at 9 weeks are showing positive. So if you're struggling to be sure of your virus infection, you could always take samples from the roots and have a high likelihood of getting an outcome that's predictable of whether the plants are infected or not. So let's see. We also looked at cross sections of leaves of stems that we had inoculated. These are healthy stem cross sections, and at the bottom are infected stem cross sections. And we found some differences in the developmental expansion, the width of the phloem and the xylem. So here in blue between the two pink regions is the phloem tissue. In the healthy tissue, it's not as wide as in the infected tissue. And you can also see that the phloem parenchyma, which, not the parenchyma, the cambial layer, which is dark pink here below the phloem, and xylem elements below that. There's a lot more lignin and cellulose deposition in the infected tissue, and along the xylem and the cambial tissue. So there's some changes in architecture that, or in cellulose deposition, that we want to investigate a lot further using a lot more detailed studies. You can see in both the cortex, the sclerenchyma. Sclerenchyma is also different, where it's a lot more hollowed out in the healthy tissue, and it's almost entirely closed in with in the infected tissue. So again, we're just starting these studies on cross sections to look at how virus is affecting developmental changes in these plants. We also see differences in the distribution of dye in infected tissue. We see more ray cells between the xylem bundles and the phloem bundles, whereas there's a more of an even distribution. The ray cells are just not as fully developed in healthy tissue, which is just curiosity things we're not quite entirely sure of yet, how the virus is affecting vascular development. But we do know that the regions, if we can go back here, in our infected tissue, the stem looks in the rosetted tissue a lot thicker than the stem does in the non-rosetted tissue. So we're hoping that we can use histology to understand what's changing in the development. Finally, we have used the infectious clone. We have eliminated different segments, RNA 5, 6, and 7, from the infectious clone and tracked infection over time through different branches, and found that we get sufficient infection in the absence of RNA 7. If you, let's see, in the top leaf, you get at 3 weeks, 90% of the plant's infected. At 6 weeks, about 80 to 90% in the top leaf. The secondary shoot, in the absence of RNA 7, much lower systemic infection at 3 weeks, and then it continues to increase at 6 weeks. So second movement from one branch to another branch is slowed by the absence of RNA 7. But if you, here where we have segments 1 through 5, if you delete 6 and 7, it seems to be restored. So you get movement into the second sheet. So there's some interplay between RNA 7 that's affecting virus movement by interacting with either RNA 6 or RNA 5, which is just a point of our further studies going forward from here to understand those relationships in how these segments control movement between one shoot and another shoot of the same plant. So we're establishing an experimental system using young rooted cuttings to study virus infection and investigate genetic reprogramming of vascular tissues, understanding how viruses spread into neighboring shoots by 6 weeks, and when phloem appears to restrict photoassimilate movement. And it looks like there's some cellulosic deposition of tissues in the vasculature that is altered during virus infection in stem. And as I said, we're looking more closely at RNA 6 and 7, governing the rate of virus movement between neighboring shoots. And that's just from another day. So
thank you, Jean Marie. Questions for Jean Marie, briefly? One.
Yeah.
Yeah. So with that, virus movement is very slow. It takes some 2, 3 weeks. So how you it gonna affect the transmission? Like if there are infected mites uh, on other shoots compared to the one which is infected, or they translate into like less efficient transmission of the virus from the other shoots compared to the infected one to the healthy plants?
We're not performing mite studies. We're really trying to hone in on tracking and understanding if we can have an experimental system to screen in rooted cuttings in a greenhouse for resistant germplasm that shows resistance to the virus or susceptibility to the virus, and also help industry to be able to study and validate their plants before they leave the greenhouse to consumers to know that their plants are healthy.
No, like if you interpret these results in the fields trial, definitely, I know you are not using mites for transmission, but how we can extrapolate like to the grower or to the master gardener, what we can say to them that either that will be less that they don't need to worry about if they see one shoot and they're infected and the rest are not infected, so they have a window like in case of rabies, like when the dog bite you, there are at least 2, 3 weeks window that the virus can make it to the vertebral column or central nervous system. So here we can see that there is a window rather than throwing the whole plant. They can just cut down the infected shoot and then they can still keep the plant.
Yeah, I guess that's a possibility, but honestly, I have to run to another meeting where I'm quite behind. I have a 2 o'clock meeting starting, so I'll leave you all to discuss these things in my absence and catch up with Oscar
Okay.
later.
Thanks, Jean Marie.